Other experiments we can perform with plants are the tracer experiments. In tracer experiments, we use radioactive isotopes. These are useful for tracing the movement of substances in plants. The C14 isotope is a radioactive isotope of carbon and can be used to radioactively label carbon dioxide. So any carbon dioxide made from this C14 and not from the normal carbon is going to be radioactive. If you grow a plant in an atmosphere containing carbon dioxide made from C14, so 14 CO2, the C14 isotope will be incorporated into the sugars produced during photosynthesis by this plant. So the sucrose made by a plant grown in an atmosphere containing radio-labeled carbon dioxide will produce radioactive sucrose. So you can trace these radioactive sugars as they move within the plant. They are radioactive, so they can be traced using autoradiography. So if you take thin cross-sections from a plant stem and place them on a piece of X-ray film, the film will become blackened where it has been exposed to radiation by C14. So as this radioactive sugar is moving, it will form a trace of blackened marks on the radioactive film. After this experiment is performed, you notice that the blackened regions are corresponding to the regions of phloem, where phloem is found in the stem. And this shows that phloem is in fact the tissue that transports the sugars made from photosynthesis in the plant. Other pieces of evidence are that when phloem is cut, you have a solution of organic molecules flowing out. If you provide plants with radioactive carbon dioxide, they will have radioactively labeled phloem after a short time. You can also use aphids, which are a type of insects that feeds on plants. They have needle-like mouthparts and they penetrate the phloem in order to suck out the sugar, and so on.